Okay, I hope you well since we had our last lecture. We'll continue today with evolutionary biology. And now we've seen that one of the most important ingredients for evolution to happen is genetic variation. So we want to understand methods through which such variations can now happen in populations. Because when a population has variations, then it stands a higher chance of evolving. But if the population is just similar or sort of homogeneous on a particular aspect, be it color, be it height, or anything, then chances that it will evolve are very minimal. So for us to understand this very important aspect to do with evolution, then we need to understand, to appreciate how variations develop in populations. So these variations within populations, be they phenotypic, be they genetic, they develop mainly through genetic differences that exist or occur between individuals. So in any given environment, organisms with different genetic makeups are likely to portray different phenotypes. Okay? Because they are the genes which will possibly interact with the environment and lead to the formation of a particular phenotype. So if within a population we have genetic differences, then we are likely to have differences even at a phenotypic scale. So these genetic differences then arise through several ways. Okay? And it is important that we consider how this evolution occurs, because that is the raw material on which evolution works. So these variations, these genetic variations, will occur in populations, one, through mutations. So mutations, as we know, when we looked at advanced genetics, we defined it, these are just the random changes in an organism's genetic makeup. So at times, these changes can happen just by chance. Okay? When DNA is being copied during the replication process, or through environmental factors, be they physical, chemical, or even biological. So most of these mutations, as they occur in a population, they will not have a big effect. So it means a mutation can occur, but it will not confer any disadvantage or advantage to the organism bearing that mutation. But there are some mutations which can create new traits, new characteristics in an organism. So these mutations that will create a new trait, will create a new characteristic, a new phenotype in an organism, are mutations that are occurring in regions of the DNA which code, the coding regions. So possibly then, that is a mutation which is happening in a gene, okay, within a gene or an intron sequence of the DNA. So if you give an example here, you can think of a population of mice which all have gray hair or gray fur. So if there is a mutation randomly within the mice, then it happens that any mouse that will have that mutation, then it will change its fur, its coat color from gray to white. Okay? So that will be a variation, is a different trait, is a different phenotype, a different color. But that mutation occurred randomly. It just happened by chance. Nothing created it. It does not confer any survival advantage. It's a mutation that was just random. But because it affected the region of the genome, the region of the DNA which codes for color of the coat. Okay, so it's coding for the color of the coat of the mice. Ile nywele ya hiyo panya. So it will change from gray to white. That is a variation because it is now a different color. So a mutation in this case will have now led to a change in coat color from gray to white. But most importantly, the mutation has occurred randomly. Another aspect that leads to variation is genetic recombination. So this is mixing of genes during sexual reproduction. Okay. Whenever sexual reproduction happens, the genes will combine and recombine when the chromosomes do the crossing over. Okay. So when organisms are reproducing, their genetic material is mixed in several ways. This mixture of the genetic material is now what creates this genetic recombination. So when you have even two siblings from the same parent, they will not look exactly the same because they will have inherited different combinations of genes, but from the same parent. So the mixing of genes, the mixing of the chromosomes, as they recombine during the giving birth of one sibling, it will be different from the same way the same chromosomes will be recombining during the formation of the other sibling. So the recombination is happening, but it is creating new combinations that are not present in any parent, and they will not be equally similar in any of the siblings. So the mixing of these genes, their genetic recombination during sexual reproduction, is now bringing around a new combination of genes that was not present in any of the parents, and for each of the siblings that will be born from these parents, they will be having their own independent combination of the various sets of genes that are present on the chromosomes of each of the parents. So in humans, each parent passes half of their genes to their offspring. So for example, one parent might have blue eyes, and the other parent could possibly be having brown eyes. Or maybe think of skin color. One is moja ni mweusi, labda mwingine ni mweupe. Okay? So then, the offspring from these two parents will inherit a mix. Okay? So it will be something like a combination of those two. Okay? Which means the section of genes that codes for this particular phenotype, if it is eye color, will have mixed in the offspring. Meaning the offspring carries a new genetic combination across that section compared to each of the parents. Okay? You might have, for example, a parent who is tall and another parent who is short. But when they give birth to offsprings, you realize that some of the offsprings are not tall, neither are they short, but they are somewhere in between. So the creation is a combination of those two traits. Okay, so there is a new trait, a new eye color in this case, a new height, a new skin color of the child born from two parents that had different phenotypes, different genetic makeups. But because of sexual reproduction, genetic recombination, the sections of chromosomes carrying those traits mixed. So the offspring is now carrying a chromosome which is a mix from the two parents. So it means if you look at the genetic makeup of the offspring, it is different from each of the parents, because the child is a mixture of the genetic makeups of the two parents. So during sexual reproduction as well, that is now when shuffling happens, of these genes happens. So when you are doing asexual reproduction, you will get exact copies. Whatever is growing out of asexual reproduction is an exact copy of what was planted. Ukipanda miwa, ile miwa inamea kutoka kwa ile umepanda. From a genetic perspective, it is an exact copy. But whenever there is sexual reproduction, then the genes from the two parents are combining. Okay, so that combination creates genetic variation, because now the offspring gets a unique combination of genes from both parents. Okay, so you get a unique combination. If you think of a bird population, some will be having, for example, a long beak, while others have a short beak. When these two types of birds with different beaks met, their offspring could inherit a combination of those traits. So the offspring might not have a long beak nor a short beak, but they could have a medium beak. So the parent, one had mdomo mrefu, ingine ikakuwa na mdomo fupi. But ule mtoto amezaliwa, mdomo yake ni kadri. Kumaanisha is a new phenotype, is a new different trait within this particular population. Why did it occur? Because of sexual reproduction, where now genes were combining. If it was through asexual reproduction, then we'll produce a genetic identical offspring. Kama ni miwa yenye iko na this particular phenotype, ile inazaliwa kutoka, yenye inamea kutoka kwa hiyo yenye umepanda chini ni hiyo hiyo. Because with that, there is no shuffling, there is no mixing of the genes, because you are not combining gametes from two parents. Another cause of variation within a population is gene flow. Gene flow is just the movement of genes from one population to another. So it occurs, gene flow occurs when individuals from different populations but of the same species interbreed. So when these populations interbreed, they introduce new genetic material into the new population that they are now creating. So because you are mixing genetic makeups of two different populations, you now end up with a higher genetic diversity in the new population. So the F1 generation that you will create after the interbreeding of the two different populations, the F1 generation will have a higher genetic diversity, a higher genetic variation compared to the two parental populations. Why is it that the new population has a higher genetic variation? Because you have mixed two different sets of gene pools. So the gene pool is the total genetic constitution of the entire population. That's a gene pool. So if you are combining a gene pool of people, for example, students from Malupe, then you are allowing them to go and mate, for example, with the students from Must. The population that comes after that mating encounter is a combination of the genes of the students from Malupe with the genes of the students in Masinde Muliro. Then now that new population will be a mix. Itakuwa na diversity, zitakuwa tofauti kuliko za wanafunzi wa Malupe na wanafunzi wa Masinde Muliro. Okay? So imagine if there is a population of wolves in one forest, and if at all they have gray fur, just like what we saw in the earlier example of the mice. Then there is a group of wolves from a neighboring forest. Okay? And these wolves in a neighboring forest have white fur. But now they migrate into the first forest. All of them are wolves, meaning they are from a common species. When they migrate into the first forest, they interbreed. The phenotype here is color of the coat color. Ya kwanza ilikuwa gray, hii ya pili ni white. So the offsprings that are coming up of this interbreeding of these two wolf populations might now be a mix of the two. Okay, so one was gray, the other one is white. So you might realize that you now come up with a mix of those two fur colors. So unazaa watoto wenye sio gray na wenye sio white, is something in between. So the difference in the coat color in the new population is as a result of the gene flow, the coming in of these wolves from the neighboring forest into the first forest. And as they come, they are coming in with their genes. So their genes are flowing. Okay, their genes are flowing. That's why we are calling it gene flow. The movement where the genes are moving along. But as they come in now, upon interbreeding, they create a new population, possibly with a different phenotype. Okay? You might have seen someone, a man or a woman who is dark in skin color. But akioa, if he or she gets married to a lady or a man who has a different skin color, wale watoto wanazaliwa, you might realize ngozi yao inakuwa tofauti. Kama baba alikuwa mweusi, unaona watoto wanaanza kuwa weupe. Okay? So that is a variation that has come because of the mixing of these two sets of genes. Another cause of variation within a population are environmental factors. So we say the phenotype you see is an effect of the environment and the genotype. So which means the environment can affect the genotype to produce a different phenotype. Okay. So for example, if you have a set of organisms within a cold climate, and these animals within now that cold climate, they end up having either very thick fur or fat layers, so that they can be enabled to survive within that cold environment. Okay? So which means through natural selection, the environment which is cold is what has made them to develop this thick fur. Ikue either kama the merino sheep. Okay, you look at the merino sheep, then you look at the sheep we see locally here. So the merino sheep has a very thick layer covering its skin. But these other ordinary sheep that we find in our areas here, unapata kwamba ngozi yake inafunikwa tu na hair. Why? Because of the environment. The merino sheep huwezi ipata huku, inapatikana in the cold environment. So in Lemuru, okay, as you go to those cold parts of our country, that's where you find the merino sheep, which has a lot of wool on its skin. But as you come to these areas which are not very cold, zile kondoo unapata haziwezi kusurvive. They cannot survive if they have so much wool present on their coat cover. Why? Because of the environment. Same sheep, but because of environmental factors, in this case, temperature, the phenotype, the animals end up appearing differently. Why? So that they are able to survive in the various environments. So with the environmental factors, it is what is present in the environment that makes the animal to change so that it can survive. Okay, so the animal is trying to change so that it can enhance its survival. It can benefit more from that particular environment. Okay, so it means then, if we change this sheep, utoe hapa upeleke, utoe merino huko ulete hapa, za kwanza zitakufa, but there are some which will survive, and with the time you will end up with a merino sheep that is not as woolly as the one that is present now in the cold parts of our country. So the environment will have now played a role to bring in a difference, to bring in a variation in the way these organisms are appearing. Okay? So the traits that are becoming more common within a population in a particular environment, those are traits that are making that particular organism or a population to survive better within that environment. We look at natural selection deeper as we proceed. The other factor that leads to the occurrence of variation is genetic drift. So genetic drift is the random changes of the gene frequencies within a population. We are saying is random, so which means they are occurring just by chance. So the trait is not helpful in any way, but the changes are occurring just by chance. The effect of the random occurrence or genetic drift is felt highly when the population is small. So when you are dealing with a population that is small in numbers, then you are likely to have a higher impact of genetic drift within that population. Okay, so with genetic drift is just the sudden change in the frequencies of certain alleles or certain genes within a particular population, only by chance. You cannot explain. They are not conferring any survival advantage. They are just changing by chance. Okay? So if you have a small group of animals that get separated from the main population, there is a very high chance that there will be random changes within this small group of population because it is small. So anything, for example, if you have a fire, moto ikuje itachoma hiyo dogo, and it might burn a section of this small population, then as the ones that are burned, the ones that die, their genes, the genes they carry will not be transmitted to the next generation. Okay? So it means then the ones that survive, the traits they carry will now be more common in the next generation. The ones that got burned and the ones that survived the fire, there is no trait that made them to survive. It was pure chance. So let's look at this example. Suppose there is a small group of birds which have long tails. And these birds with the long tails are separated from the main population by a storm. So kuna upepo ama nini kuja inafanya zinakuwa separated. So over time, this isolated group might have more birds with long tails, even though there was no real advantage of having long tails. What led to the separation? What led to the isolation? A storm. A storm occurred just by chance. And there is nothing that makes the phenotype of these birds advantageous. Okay? So the long tails don't confer any survival advantage. But they are likely to continue, for example, having those long tails. Pure chance. Pure chance. No advantage. that is being conferred by the trait that these birds carry. So to put it all together, variations within populations. Remember we said evolution is about populations. So within populations, variations will occur. Variations will arise due to mutations, genetic recombination, sexual reproduction, gene flow, environmental factors, and genetic drift. Okay, and genetic drift. So these variations, the variations within the genetic makeup, the genetic constitution of these organisms within the populations, are what will now give the organisms that make up those populations different traits, different phenotypes. And those phenotypes are now important for evolution. Why? Because they provide alternatives, they provide options. Okay? If, for example, you are having differences in height, or differences in skin color, or differences in resistance to a particular, susceptibility to a particular drug. If you have bacteria, some are resistant, some are susceptible. When that drug is used, the bacteria which is resistant will not die, okay, will not be killed, but the ones that are susceptible will be killed. So in essence, the ones that could withstand that drug was because of the variations that occurred in the population. And it is because of that variation that the entire population was not wiped out. So evolution has happened. Why? Because of the differences in the traits. Okay? Because of the differences in the traits. Under genetic drift, there are three important effects that we can look at. These effects make up what we call the three concepts of genetic drift. Genetic drift, we said, is the random change in allele frequencies within a population due to chance events. That is genetic drift. And the three main concepts of genetic drift, one is found as effect, another one is bottleneck effect, and lastly, what we call the Warholund effect. So these three, because they are concepts of genetic drift, and we've seen that genetic drift has a higher effect, impacts smaller populations more, then it means that these three will help us to be in a better position to understand how evolution works in small populations. So let's try now to break each of these three concepts down. And we'll begin with the founder's effect. So the founder's effect occurs when a small group of individuals from a larger population establishes a new population in a different location. Okay? So in a way, it means there was initially a parent population. Then a few organisms from the parent population moved away from the parent population and established a new population, most importantly in a different location. So one thing is, the population that moved away from the parent population ilikuwa ndogo. So it is small. That the founding group is small in size. Meaning the genes they carry, they are also smaller in number. So the different genes that the small population carries within them, you cannot compare. What we call the genetic diversity of that small population is smaller compared to the parent population. So it is possible that some genes, some alleles may be either overrepresented or underrepresented compared to the parental population. So in this, the example we have here, they are butterflies, the ones that have moved. In the first instance, we have butterflies of a certain color. In the second instance, we have butterflies of a mixture of those different colors that are present in the founding or the parent population. In the third instance, we have still butterflies just of a similar color, but that is not the only color from the parent population. So over time, when now these populations that moved away, these founding populations, when they keep on reproducing, when they keep on producing new populations, each of them, because it is carrying a different genetic diversity compared to the parent population, it is likely that the populations that result from them will have different genetic traits compared to the parent. Because the small population has less genetic diversity, the only expectation is the populations that will now continue to come from these founding populations will still have a smaller genetic diversity compared to the parent population. So founder's effect, in a way, then leads to a decrease in genetic diversity of the new population compared to the founding population. How was the choice of the founding members random? What is the effect? There is a change in the frequencies, what we are calling genetic drift. Why do we have that change? Because only a small population, a small group moved from the parent population, isolated itself, and began a new population. Okay, so in a way, founder's effect leads to a reduction in genetic diversity. Okay, founder's effect will lead to a reduction in genetic diversity. Let's proceed. And now the key points under founder's effect is: one, you have a small isolated group that is forming a new population. It's important for the word isolated, so there is no gene flow between the parent population and this small group that has moved away. Okay? So the new population that is forming from the isolated population will now have a smaller or a genetic diversity that is either smaller or lower compared to the original population. Okay? So because of that, you end up now having some traits being more common or rare within the new population compared to the original population. Why are they common? Why are they rare? By chance. The formation of this small group happened by chance. So the genes they carry, the traits they carry in the new group that is resulting from the small isolated group is as a result of a chance event. Okay? It's as a result of a chance event. The example that is given, we have within the United States a population referred to as the Amish population. This population is a group that originated from a small group of Swiss German founders as far as the 18th century. So because the group that created the Amish was small, okay, it meant that it had limited genetic diversity. And because of that isolation, there was no gene flow between this small group and the Swiss and other German populations. The resultant groups have also, the resultant population from it have resulted in a limited genetic pool, which now has some certain genetic disorders. Okay, so there are some rare forms of dwarfism, which is far much more common among this population than when you compare from the parental population that it came from. Okay, so the disorder was present in the original founders, but it has become more prevalent in the isolated community due to the founders effect. Okay, so with founders effect, you have a population that is isolating itself from a parental population. As it isolates itself, as it moves away, it carries just a tiny amount of diversity, a tiny amount of traits from the original population. Then now, as this isolated population continues to reproduce, continues to interbreed, it will bring new populations that have just a lower genetic diversity compared to the parent population that the isolated group came from. Okay, the reduction in the genetic diversity will mean some traits, some phenotypes in the new population will either be rare or more common compared to the parental population. Let's now move to the bottleneck effect. So, whenever the size of a given population drastically reduces due to a catastrophic event, then we can refer to that as a bottleneck. So possibly there is a disease, there is a fire, or people fight like what's happening now in DRC. In the process, some form of life is lost. So there are trees that will be burned, there are people who will die, there are some animals that will die in the process. So once the animals are dying, once people are dying, once trees are dying, we are leading now to a drastic reduction in the size of that particular population. If we are talking of trees of a particular species, it will be a reduction in the population size. If there is a drought, for example, in the northern part of Kenya, then sheep die. That is a bottleneck effect on the sheep because their population size has been reduced drastically as a result of a catastrophic event. So in the event that only a section died, the surviving individuals will be having a limited genetic diversity. So maybe there were 100,000 sheep. Out of the 100,000, let's say because of drought, 40,000 zikufe. What remains is 60,000. If now the 60,000 have survived and they have to reproduce so that the population recovers, you cannot compare the genetic diversity of the 60,000 compared to 100,000. Okay? Because the ones that died will no longer participate in the formation of the next population. So in a way, they died and now they are not able to be to pass their genes to the next generation. So then it means the populations that are coming after the bottleneck event will have a genetic diversity that is low compared to the initial population. Okay? So that's why we are saying here that it will result in the loss of certain alleles. The alleles that were with the 40,000 which died were lost. So the new population will be having genetic traits that are very different from the original population. So in a way, a bottleneck event could either be death or even there is an earthquake that separates populations. Okay? So hii imeenda huko, hii imeenda huko. The genes they've gone with are different. The genes that have gone this side are different. So as the two populations begin to try to recover, the diversities that they will have are different from the original population. When you look at the photo on page 6 of the bottleneck effect, so it is a chance event which greatly reduces the size of a population. Think of a fire, think of an earthquake, think of war, anything that leads to a reduction in the population size. So the initial population before the incident had, in this case, a mix of both red and yellow alleles. But if there is a reduction in the size, in the population size of those organisms within now the population, which is the body of this bottle, then whatever is left, whatever survives after the reduction, after the chance event that led to the reduction of the population size, will likely have allele frequencies that are different from those of the original population. But we now have to create a new population from the surviving individuals. We have to create a new population from the surviving individuals. So it means we've lost some alleles because of that chance event that led to a reduction in the population size. Some alleles were lost when these animals died, when these plants died. Okay? Some alleles went with them. They will never, those animals that died will never participate in the regeneration of the new population. So it means then the alleles that are carried by the surviving individuals are different. The frequencies that they have, those alleles within the surviving individuals, will be different from the frequencies of the same alleles in the original population. So when we begin now to create a new population from these surviving individuals, the new population that we are creating will be dominated more by the alleles that are more within the surviving individuals. So then you create a population, a new population that has a different allele frequency compared to the original population before the bottleneck event. Okay? So with bottleneck effect, the key examples, the key points to note is that there is an event, a catastrophe that leads to a significant reduction in population size. Okay? So the population is not decimated in total. Hawakufi wote. Out of maybe 100, they could survive like 40 or 50. Okay? So what happens now, because of the reduction in the population size, the ones that survive, they will eventually have a different allele frequency compared to the initial population before the catastrophe. So when now you begin to use the surviving population to create or to regenerate a new population So the individuals that survived are less compared to the initial population that you had. Meaning the few who survive must be having fewer allele frequencies compared to the many who were there before the catastrophe. So as you begin now to reestablish your population using the survivors, then the new population that you get will have genetic diversity that is lower compared to the population before the occurrence of the what? Of the catastrophe. Okay? So that is a bottleneck event, a bottleneck effect. It can be, if you are dealing with the genes, then it can be a genetic bottleneck. Okay? It can be a genetic bottleneck. A well-known example of the bottleneck effect occurred with the cheetah population, the population of cheetahs. So many years ago, thousands of years ago, there was a significant change within the environmental conditions. And that event led to a dramatic reduction in the cheetah population. Okay? So we had so many cheetahs that died. So the ones that were left zilikuwa ndogo sana in number, significantly less. Okay. The surviving population was made up of a cheetah population with a significantly lower population size compared to the population before the change in the environmental conditions. So then, the cheetah population that now had to be created from the surviving ones today ends up to be fairly genetically similar to one another. Why? Because the ones that died removed their alleles from the gene pool. The genotype that is being created was from a very limited gene pool. Okay? Because the ones that died could no longer participate in the creation of the new population. So the new cheetahs then were created from a very limited choice of a gene pool which had a fairly low allele frequency, which then now meant that the current populations will have a significantly lower genetic diversity. And that's one of the things that now makes them to be more vulnerable to diseases. It makes them to have a reduced ability to adopt given the environmental changes. Because with a bottleneck effect, you are reducing the variation, you are reducing the diversity. And we said for there to be evolution needs a high diversity. Evolution needs variation. Okay? Evolution needs variation. But a bottleneck is reducing the variation. So the last concept in genetic drift is the Wahlund effect. Under the Wahlund effect, the example I will give, think of it, you finished school, at the end of these 3 months, at the end of this semester, you will be done with your degree. Then maybe you've been harboring interests kwamba unataka kufuga kuku. So you will go home, then maybe you want to buy 50, uanze na vifaranga 50. So when you get someone who is supplying you, unapata labda mmoja ako na vifaranga 10 tu, lakini wewe unataka 50. So utachukua 10 kwa huyu, uende tena utafute mtu mwingine akupee, upate labda ako na 25, uchukue, mwingine akupee, mpaka uweke pamoja zifike 50. But the most important thing you need to remember is you are taking 10 from one person who has a bigger number of birds. So maybe ako na 100 lakini zenye anauza hiyo wakati ni 10 tu. Mwingine naye mwenye wanakupea 20 maybe ako na 200. Ule mwenye utaenda upate 5 maybe ako na I don't know how many. So you end up now just bringing together sub-populations. When someone gives you 10 na ako na 50, that is a sub-population. Umechukua tu a number of the population that person have. So you are bringing together different sub-populations to create your 50. To you, it is one population now, but the sources are different. So remember, the subpopulations that you are bringing with will be having different allele frequencies. Okay? There will be a different allele frequency. So the population you are creating is a mix of different subpopulations. When you compare the parent populations where you obtained your subpopulations from, the parent populations will likely have a higher diversity because the alleles you are obtaining from each of those farms that you are getting your subpopulation, the alleles that you are taking are just a tiny representation of what the entire population has. So the ones you are creating, the population you are creating by mixing different subpopulations will eventually have now an overall reduction in genetic diversity compared to the different source populations. Okay? So with Wahlund effect, wewe unataka kuanzisha a common population. But for you to get to 50, huwezi pata hizo 50 from one person. So you have to pick them from different populations. You are thinking that you are now increasing, you are getting animals that are unrelated. So when you bring them together, you are likely to have an increased, what we call hybrid vigor in agriculture. But when you compare your new population with the different parental populations, you realize that your new population will have a relatively small genetic diversity compared to the original populations. Okay. So this effect is often observed when the populations that were originally separated come into contact and interbreed. Hizo kuku zenye utatoa kwa hizo from different people utaweka pamoja, then kutakuwa na jogo na kutakuwa na hiyo kuku kike. Zitaanza ku interbreed na zitage. As they interbreed, they are mixing different gene pools. Okay? And you are now creating a reduction in diversity compared to the population that you obtained from. So in a way, you end up with what we call inbreeding depression. Okay? So you are reducing the diversity. And even if the populations that you've brought in were not related, which meaning there is no inbreeding because you are bringing in animals from different farms. But you end up with a reduced heterozygosity. You end up with a reduced genetic diversity. Why? Because you are mixing genetic alleles from different source populations. The subpopulation you are picking from each of the source populations has a reduced allele frequency. So when you compare the allele frequency within the new population, it will be different from the allele frequencies in the source populations. The difference is the new population will have a reduced heterozygosity. Okay? Why? Because you mixed subpopulations having different allele frequencies. Okay? So that is the Wahlund effect. It is almost similar to the founder effect, only that in the founder effect, the founding populations are not being mixed. They remain isolated. But in the Wahlund effect, the population you are getting from farm A, in the Wahlund effect, you are bringing it together with a population from farm B. Then you go to farm C, get a subpopulation, you come and mix. So you have a new population of subpopulation from farm A, farm B, and farm C. Different allele frequencies. Eventually, you will get a population with a reduced genetic diversity. Even if the source populations were not genetically related. Okay? So that is the Wahlund effect. You obtain organisms from different populations, then you bring them together. Then as they interbreed, you lead to a reduction in the genetic diversity, a reduction in the heterozygosity. Okay? So the examples that are given are just trying to explain what I've put across. Okay? So that is a summary of those three concepts within genetic drift. In the founder's effect, a small group breaks away from the larger population. Okay? And as it breaks away, it leads to the creation of a new population which will have a reduced genetic diversity compared to the source population. So with the founder's effect, there is nothing like a reduction in the numbers. It's just an isolation. Okay, so you think of it, if you have animals moving from this park to a different park, okay, the animals that are moving, sio zote from park A, is just a small group from park A to a different place. Then they will begin a new population in their new environment. So the new population will end up having a lower genetic diversity compared to the source population. In a bottleneck, there is a drastic reduction in the population size, mostly due to a catastrophic event. That reduction in population size means the ones who die as a result of that catastrophe will not participate in the creation of the next generation. So only the surviving individuals, which will be a significant reduction compared to the original population, only the surviving ones will be used to regenerate the new population. And because only a few have survived, it means the alleles that went with the dead ones will not be part of the new population that is being regenerated. Okay, so many alleles were removed from the population as a result of the catastrophe, meaning the new population that is being generated will be having a drastic reduction in the genetic diversity. In the Wahlund effect, you have the mixing of genetically distinct subpopulations. We gave an example, unachukua kuku kutoka kwa shamba tofauti na unaleta pamoja. Okay? So when you eventually do an analysis of the diversity within the new population, it will be reduced compared to the parental populations. So in a way, the Wahlund effect will then mimic inbreeding. So you are likely to begin experiencing inbreeding depression within that particular population that you've brought together from different source populations. Okay. So these effects all show how genetic drift and the changes in population size, structure, and mixing can significantly influence the genetic makeup of a population, leading to evolutionary changes. Okay? So because of founder's effect, because of bottleneck effect, because of Wahlund effect, all these are dealing with a small population because they are concepts within genetic drift. They will lead, they will influence how the genetic makeup of a population is changing. And once the genetic makeup of a population has changed, then we are likely to have evolution. Okay? So those are the three concepts within genetic drift. We also saw earlier how variation within populations arises. Okay. So in this section we've seen how the variation, how the differences occur within a population. In evolutionary biology, it's important to understand the occurrence of differences, the occurrences of variation, because it is on the basis of that variation that now evolution works. It's on the basis of that variation that evolution works. Okay? So I'll stop there, then share this recording. I request that you go through it, and if you have any question, you can reach out. You know my email, which is at the bottom of each of the pages of the notes that I'm sharing. Okay? So go through. If there is a question, I'll be happy to answer. So thank you very much, and I'm looking forward to seeing you in our next class. Thank you.